welcome everyone to the second masterclass organized by the EPIC Butterfly Project. This masterclass is about Atlantic butterfly species, and the person that is going to give this masterclass is Chris van Swaay. Chris van Swaay, many of you will know him, but for those who doesn't know, Chris is a biologist specialized in ecology and conservation of butterflies, particularly in the Netherlands, but also in the whole Europe. He's has been coordinating the Dutch butterfly monitoring scheme since the 90s, has published extensively about butterfly trends, first read that data book of European butterflies, book of prime butterfly areas in Europe, updated red list of butterflies in Europe, etc. All this work has done it through Butterfly Conservation Europe, which is also who is also part. Chris is also part of this institution, and also has brought together data from different monitoring schemes across Europe to produce the European Grassland Butterfly Indicator, as well as a European Butterfly Climate Change Indicator. So most of you probably will know him because of these very important works. Well, I think that's all about Chris. Is perfect. Is the perfect person to give this this presentation. Just as I share in the chat, this presentation will last 1 hour. You are all welcome to ask whatever you need. Any doubt should be pasted in the Q&A section, and every so often I will go, and if we see we have many questions, I will stop Chris and we will solve the questions. But the idea is to cluster the questions and try to answer them every time Chris goes from one family to other family of butterflies. So I think that's all. The floor is yours, Chris. Thank you.
Yeah, thanks. So welcome. I will give a talk about, I think about an hour on the Atlantic butterfly species, but only the difficult Atlantic butterfly species. Nothing happens. Yes. So it's good to realize that the Atlantic is actually the very western part of the continent. So actually it's this little bit on the edge going from Norway over the west of Denmark, over the northwest of Germany, the west of France, the west of the Benelux, north of Spain, and the north, very north of Portugal, and plus UK and Ireland. It's good to realize we are not going into the heart of the continent, and as a consequence, we have not that many species on this part of Europe compared to, for example, the Alps. Or last week was Rudy in the Balkans. That's many more species. So we have about 216 species occurring in this bit of Europe. And about 182 have more than 1% of their distribution in the Atlantic, so that you can really call them Atlantic species. Now, I also skipped the Picus d'Europa because actually they are more an alpine butterfly and it would generate a lot of extra species. So I talk mainly about the whole Atlantic, but the Picus are a bit on the edge. It's not clear. So one of the lucky things we have, and I will show some distribution maps regularly, is that we have a lot of species which look like each other, but they don't overlap in distribution, which makes it much easier to identify them. And I'm not giving this talk on my own because I use a lot this book by Peter Gergely from Hungary. He wrote this photographic guide. You can download it, but of course it's a bit too quick to type that. But in theory, if you Google for Gergelian butterflies in Europe, you should be able to find the PDF. And that's, I really like this book. It has a lot of photos and always with clear help on where you should look at. So Peter made this book. You see a photo of him, and it covers most of Europe, including most of the Atlantic. And it has, so like I said, a lot of these small arrows and a text pointing to where you should look for the differences between the species. And this is really a helpful book. I use it a lot in the field, and I can advise everybody to have a look. So now we start with the group. The first group we talk about is the Hesperiidae. In Europe, you have two groups, two genera of brown, dark brown Hesperids. You see them here. On the left is the Spialia group. In our region, it's just one species, Spialia sartorius, but in Spain, you also have Spialia rosea if you go further south, and on the Balkans, you have other Spialias or Bifer and others. But in the Atlantic, it's actually only Spialia sartorius. And you can recognize it by these four white spots in the top of the wings, of the forewings. And look at the right, you see that Pyrgus always has three spots on that point. And that makes it relatively straightforward to identify Spialia from the Pyrgus. But the Pyrgus also have some species. The main species you will see is Pyrgus malvae, which can recognize on this typical, on the upper side, on this very typical, we call it always tooth-like white marking that you find on the back wings. Both Pyrgus malvoides and Pyrgus malvae have that, and this species can actually, well, not reasonably be identified in the field, only by DNA, and they exclude each other. So if you're south of Bordeaux, you probably will have Pyrgus malvoides. If you're north of Bordeaux, it will be Pyrgus malvae, and it's a species which goes all the way up to the north of Denmark. So it's a species well distributed in the Atlantic zone. The other important species of Pyrgus we have in the Atlantic is Pyrgus americanus. This is a species which is expanding. So at the moment, the main distribution is in France and Iberia, but it's really moving north. And you see here the first edges where it's coming. And well, we don't know for sure, but it looks like it will start colonizing parts of the Benelux and northern Germany in the next few years. So it's a species to watch out for, to take care. It's a bit bigger, a little bit bigger than Pyrgus malvae, and you can recognize it. Now this is just a second. Now I can see my own screen. You can recognize by this typically shaped, arrow-shaped white spot in this part of the underwing. And also this with red indicated white area is quite different between Pyrgus malvae, the other widespread species in most are malvoides in the Atlantic, and Pyrgus americanus. In the field, it's relatively easier because americanus is bigger, but of course, if you don't know the species and you see it for the first time, it can be a bit puzzling. But once you've seen several malvaes and you see an americanus, you usually know directly that at least you see something different. The other group which can cause problems are the yellow Thymelicus and Ochlodes and Hesperia, so the yellow Hesperiids. For people who find these two species, Ochlodes sylvanus and Hesperia comma, they can be quite alike, especially old Sylvanus, these dots can be much whiter and look a bit like these ones. But Hesperia comma always has these white dots on the underwing, on the basis of the underwing, which Sylvanus never has. It's always uniform. The upper side is also a little bit different. And with the males, it's most easy because this is a male androconial gland. And the shape in comma is a bit triangular with the biggest, it's widest at the base. And with Sylvanus, it's widest in the middle. But there is another thing that Sylvanus is relatively early in the season. So it flies from mid-May to the end of July. And in most areas in the Atlantic, Hesperia comma is relatively late. So it starts mid-July and flies until September. In the mountains, though, it can be different. In the mountains, comma can be earlier. But we're talking about the Atlantic today, and there already the time of year helps to know which species you are looking at. So in this case, to the left, you see a typical Ochlodes sylvanus in June when they are fresh. The other difficult species are the Thymelicus species. Now, this, of course, is what you read in almost every book. The underside of the antenna clubs is yellow in Sylvestris and black in Lineola. And when you are almost lying on the ground, you might be able to see that. So these photos show that indeed the underside of the two species are different. But very often you don't get a chance. These are very fast butterflies and they can be very hard to see this way. So what should you do? The most important thing is to remember that Thymelicus sylvestris is relatively early, is 2 or 3 weeks earlier than Thymelicus lineola, which means that if you go early in the season, you find mainly males. And this, again, this androconial patch is different. In lineola, it's relatively short and parallel to the veins. In Sylvestris, it's much longer, much clearer, and not parallel to the veins. This makes it so the males are much easier to identify than the females because, well, formally, if you measure them, I think Sylvestris is 1 millimeter bigger or so, but you won't notice in the field. So they will fly mixed up and you don't have time to see that. But this, with the males, it makes it very easy. So try to be there on time if you think you might see this species. Thymelicus actaeon is the third species of this group. You can see it's a typical species in most of Central and Northern Europe. It's typically a species of calcareous grasslands. And in many parts of the Atlantic it is, but it also occurs in other habitats, so you should not completely focus on that. Very typical with this species is this band of light spots on the upper side. It's not so clear in the males, but here, for example, in this female, it's very clear. And it's also clearly smaller than the other two. Now, the other two are also small, but this one is really small. So it's really a small butterfly, and it can be a challenge to identify it. So take care, take your time, because they can be really fast. Okay. Juan Pablo, do you want to other questions? Because we are now through the Hesperiids.
No questions so far.
Okay,
So
then we move on to the Pierids. Well, the main two groups to identify is actually the Croceus group, which is on the upper side is orange, and the Hyala alphacariensis group, which is on the upper side yellow or whitish. Females are whitish. But usually they will never show their upper side, I mean, unless they are flying. So the first thing you have to do is check if you can see the upper side when they fly. Is it really orange? Then it's all as good as always Croceus. Is it yellow or whitish? Then it's one of these. But if they sit down, many people find it very difficult to identify them. Now, it's not that difficult because on the underside of the forewing, Croceus is orange. It has more or less the same color as the underside of the underwing. But with Hyala alphacariensis, this is, well, females is anyway clear because they are much lighter, but even the males, they are yellowish and not orange. And they are a bit lighter than the underwing. So there is a contrast between the underwing underside and the upper wing underside. And in Croceus, that's much less the case. Once you have seen this, it makes it relatively easy to identify them in the field, at least the two groups. Because Hyala and alphacariensis, well, I think they cannot be identified reliably in the field. You have to either kill them and use DNA. But the larvae are very different, and also the food plants are very different. So Hyala is typically a migrant from Central Europe, and it uses for the food plant mainly all kinds of clovers and other legumes. Alphacariensis is more or less always on calcareous grassland. It's much less of a migrant. And it feeds on species like Hippocrepus squamosa and Coronella. And so it's a very different habitat where you find them. But also Hyala is typically found, let's say, in the northern half of the Atlantic. Well, this looks like it's a Dutch species, but that's because there are many records from the Netherlands. But it's you find it also in northern Germany and the northern part of France. It rarely gets to England. I think it's really rare in England. So they are coming mainly from Poland and Russia and Hungary. So it's a Central European species migrating west. Alphacariensis is this species. I think its populations in the Atlantic are all south of the River Seine. So it's in the central and southern part of France and the north of Iberia. You can find alphacariensis. So that helps to identify. If you're really in the very south of France and you see one of these yellow ones, it is almost certainly alphacariensis. We have another problem with the females of Croceus. They can be of the form helice, which is whitish. So instead of being orange, they are have the same color as females of the Hyala alphacariensis complex. And of course, they always sit with their wings like this, so folded. The easiest way, there are two things that can help you. First, if the light comes from the other side, you can see these yellow spots in the back of the wing, which are always missing in Hyala alphacariensis because they only have a small black edge. But with Croceus helice, it's much wider, and so you can see these yellow spots. And also this is much larger, so there's a lot of black on the upper side, where with Hyala alphacariensis, this is a relatively narrow edge. But they can be difficult, if certainly because these Coliases have the habit of flying very fast, racing over your field, and then it can be a challenge to have a good look at them. But still, especially the females, do sit down to drink, and that makes it easier. It's especially the males, which can be flying endlessly. The whites. Now, the whites used to be relatively straightforward in the Atlantic, but now we have 4 species of Pieris whites, which we have to take care of. First of all, the large white, Pieris brassicae, it's much bigger than the other 3 species, and the black goes all the way down, so it has a huge black apex, so the point of the forewing, a huge middle spot, and this is very typical for Pieris brassicae. Now I skipped the one, but I go to Pieris because this is now probably one of the most abundant whites everywhere in Europe and also in the Atlantic. Pieris the small white. It has a black apex, but you can see it doesn't go down as far as with Pieris brassicae. So it's only in the top edge of the forewing. I've also pointed that out here. And it never reaches the same vein as, or never, almost never, reaches the same vein as the middle spot. Now, Pieris is the new species, the new kid in the block. It arrived in the Netherlands around 2015, and now it's to be found everywhere in the Netherlands, in Belgium, northern France, northern Germany. I don't think it has reached Denmark by now, but otherwise, probably it soon will. I know there are good populations near Kiel, which is not far south of the border. Anyway, so it's a species the Danish can expect, and it's also one was found in the UK last year. This is a very typical example. So the black in the apex goes way down up to the same vein where the middle spot is. You can see it also here. But a very important, the middle spot is not round, but it's always convex to the outer edge. So look here, Rape is round, Brassicae is round. This one is completely the other way around. Of course, this is a very nice example. It's not always that clear. But anyway, it's a small butterfly with a large black spot. And once you notice, and like I said, there is often a connection between the middle spot and the outer edge. You can also see that here. That's also important. Yeah, it is. The problem is a little bit that in spring they are not this clear. This is typical summer form. In summer they are relatively easy because everything is very pronounced. But in spring, this can be less pronounced. It can be a bit more difficult. If you are in the field, what helps? I always have the feeling it's a bit smaller and more rounded than Pieris rape. It always reminds me of a Leptidia a little bit, the way it's flying. It's a little bit different way of flying, and then you can recognize it already from a distance. And the green veined wine Pieris napi is the last species, it has this typical on the upper side. Of course, the underside is very different in this case. It has these green veins. But if you only see the upper side, this goes like a little bit, this black goes in very small portions along the veins, along the outer edge. Now, this could resemble this species, Pieris mani, but in this case, the middle spot is always rounded in Pieris napi, and even often really rounded completely, very clearly different from the convex middle spot that you find in Pieris mani. Okay, the Pieris species. Questions? No questions?
Not so far.
Okay.
Everything is very clear, Chris.
Thanks. So we go to the blues, the Lycaenids. We start in the beginning with the Satyrium species. They can be difficult to find, and they can be difficult, although some of them can be in big numbers. But still, I know that many people find them actually quite difficult. So let's start with the easy ones. W album already says it has a white W, and you can recognize that here. So this is a species always bound to ulmus trees. So you will always find it on or very close to ulmus trees. That is already very helpful to identify the butterfly. It's always typically, I think it can complete its complete life in the tree. So It lives as an adult in the tree. Eggs are in the tree. The larvae feed from the leaves of the tree, and the pupae hang on the tree. So this is typically a species which does everything on the almond tree. Sometimes it cannot be that clear, the W, you get something like this, but still this, yeah, once you know the typical orange band, it's W album. Prunus is typically a species of Prunus spinosa hedges and things like that. Has very much orange. You can see this. This is really much more than most species, the other species have. And it's actually very, very clear usually in the field. Spiny has a large blue spot on the outer edge of the hind wing. Spiny can be also in bigger numbers. Sometimes you see them in groups. So, and these can be the more difficult ones. Satyrium illicis and Acaciae. They can be really hard to identify if you see them for the first time. Illicis has the more clear white band compared to a relatively unclear band in Acaciae. Also, both can have a small dot, but in Acaciae, a small blue dot, sorry, like here. But in Acaciae, this is usually more clear. So you see more clear blue dot. So, but an Acaciae is really small. This is really a small butterfly. So in the field, that also helps. But if you just have a photo, it can be tricky to know for sure. Other blues which can be difficult, is the group of the Cupido species. These are all small or very small species, blues. I added Cianeri semiagus on the right because it is clearly bigger in the field, but the underside looks like the others, so that's why I included it too. Now, Argynnis is a very, very widespread species which is expanding north very quickly. It has a very clear tails on the wings, very relatively long and very clear to see. So only if they are fresh. Of course, in old specimens, this can be more difficult. And it has clear orange dots, usually. If old specimens, they often lose, or this orange gets fainted, so it gets more difficult. But then old specimens are always difficult. But you see, this is the most, in the Atlantic zone, the most abundant other species. The tails are shorter. It normally doesn't have any orange, and it has these typical black markings here. So that's Alcetas, which you can see, especially in the south, the sun half south of the Loire, as I say in the text. So it's not in northern France or the Benelux. Now, Osiris and Minimus, they can be difficult, although Osiris in the field is clearly bigger. Minimus is a really very small butterfly. You actually always find it near or on Antilles species. Here you see typically what they look like, these plants. So they're always on or very close to Antilles plants. The same applies to Osiris, which is always close to Onobrichis plants. So Cupido Osiris is always, and that's also why it's very much linked to chalk, because Onobrichis is a plant you find on chalk. Cianea semiagus you can find on many more habitats. Usually the larvae feed on red clover, and it's actually quite widespread, but not in the UK and Ireland. These are typical photos which you see of two of these species. So Osiris is typically on Onobrichus, which looks like this. It's a pink flower. Once you recognize it, it always helps to find butterflies. And this is typically Cupido minimus, Ona antilis flower. They do everything in the flower head. So the eggs are laid, the larvae feed here. So this is very much, and you can also see how small it is. It's hardly any bigger than the flower head. Small blues. The Phengaris or Maculinea butterflies, they are typically species which are large. So in the field, certainly Phengaris arion or Maculinea arion is a big butterfly. One of the biggest blues we have in Europe. On the upper side, they are all blue with some black dots, but they usually don't show the upper side a lot. These butterflies typically like to sit and fold their wings, and then you see the underside. And they are actually quite different. So Arion has huge black dots and a very clear two rows of dots on the back of this hind wing. And it's always on this type of habitats. It really likes calcareous grasslands. It feeds on thymus and origanum, which you also find in dry situations. Alcon and Thales can be very hard to identify. They are a little bit different. You can see the way the black dots are arranged is a bit different in Alcon than in Thales. These can occur together in some parts, but I think it has now become very rare that you find them together. I've personally only experienced that a few times. So usually you only find one of them. Both Nausitaus and Thales are typically bound to this food plant, Sanguisoba officinalis. So you will always see Sanguisoba in the neighborhood. And actually, it works the other way around. You drive on a road, and suddenly you notice there is a field with Sanguisobas because once you recognize them, they are easy to see from the car. And then you start to search for Thales and Nausitaus. Alcon, however, can also occur on these grasslands, but usually in the Atlantic is on another habitat. So you see the distribution map. So Arion is introduced in the UK. I think this is just a few sites because there must be many more. But especially in the south of France and in Iberia. Alcon you can find on quite a few places, but very scattered. Thales is really rare. You find it mainly in the Netherlands and in France. Nausitaus you can find in the Netherlands and in northern Spain. Typical habitat of Alcon in the Netherlands. You see a humid, wet heathland. This is the habitat of Alcon in Portugal, North Portugal, where it's also a heathland habitat. And often these butterflies can be hard to find, but the eggs are relatively easy to find. They, for example, lay their eggs on Gentiana pneumonanta, and you can find these eggs relatively easy. You find the eggs often before you find the butterfly. Once you know where to look, you find, because the butterflies are often in a very low density, and they can be hard to find, where the eggs are much more conspicuous. And of course, the food plants are very conspicuous, so it makes it easier to identify them and find them. Anyway, if you do an inventory, you should always, and you find gentians, it's always worth checking in this late July if there are, or the beginning of August, if you can find eggs of Alcon. The Plebejus species can be really difficult. In most of Europe, it's actually Plebejus argus, which you can find almost anywhere in the whole Atlantic zone. Um, However, it looks quite a lot like Plebejus idas, which is the lower one here, um, which is actually in the Atlantic zone, mostly in Denmark and Norway. And there are some scattered observations south of that area, but I'm not sure if they are correct. They are difficult to identify. What helps most is in males, usually the black border of the wings, of the upper side of the wings, is very broad and very clear, dark. In Idas, this is usually much less, a bit like this. In the field, that's often the easiest way to identify them. If you really want to be sure, you have to take some home and look more closely. There are some other characteristics which you find in the book, but in the field, this can be an issue. Argyrochromon is actually a bit larger than the other two, and has this very orange underside. Very also on the underside here you find a clear orange line, but it's also on the forewing, and that's almost missing in Argus and Idas. You almost find, well, small orange dots, but here is a very dark and very clear orange dot on the underside of the forewing. That's Agrochromon. I think I have some maps. So here you see Argus comes from all the way in Norway over the UK down to Portugal. Idas, well, mainly in Norway and Denmark, maybe northern Germany, but I think the rest, I'm not sure. Maybe in northern Spain it might be. Agrochromon is mainly in France. So in this part of France, you can bump into Agrochromon. It has also different habitats. So Agrochromon you can, for example, in France find in woodlands, yeah, on open places in woodlands, where Argus is typically a species which prefers heathlands. That's,
Chris,
yes,
sorry, there are several questions.
okay.
Let's go first with the one more related to the Lycaenids now. Is Andrew asked both questions, but Andrew asked the second question of Andrew is what is the status of Teleus in the Netherlands, Belgium?
Well, I think the species is not in Belgium anymore. I think it's extinct already quite long ago. In the Netherlands, it's still present. We have one introduced population, and which is still there in some years in good numbers. So yeah.
Okay. Also, Andrew asked about Pieridae. Hello. What is the abundance ratio of money to rape in the Netherlands, Belgium? Also, please, could you describe again the characteristic flight action of money?
Okay, I'll go back. Oh, no, too far. So first of all, most of the whites you see also in the Netherlands is rape. That's while rape and napi. They are by far still the most abundant species. But money, especially in summer, uh, can be relatively widespread. But you don't find them a lot. You find one here and one there, or two here, two there. So the numbers are much lower still than I heard that in parts of southern Germany where the species is already longer, that there the numbers of money are higher. But let's see how things develop. But at the moment, um, if you see one money, you see 10 rape and 10 uh, napi. So it's still much less abundant than the other two whites. And the characteristics, well, if they fly in the field, but this is from my own experience, they fly a little bit, they are a little bit smaller and the wings are a little bit more rounded. And it gives them another It's a bit difficult to explain, but if you see Leptidia flying, they have a very different way of flying. And it's somehow, I always feel it looks a bit more in the direction of a Leptidia. And that's what I was trying to explain. That helps in the field, but um, it is a little bit different way of flying. And yeah, you have to see some of them, and then if you watch closely, it starts. Somehow you get a feeling how to recognize them easier, because otherwise you have to look very carefully. And also the eggs are laid on the same plants, so of course money lays eggs on a limited number of plants, but rape will also lay eggs on those plants. So. Um, that's also not always helping. You can also recognize the small larvae of money. They have, oh, I always forget, they have a black head if they are very small, and rape has a green head. Or it's the other way around. No, I think it's that, like that. But then you have to rear them, for example, and you can see that. Does this help?
I think so. I think so. Let's wait for, well, there are no more questions, but I'm sure people will start asking
Okay.
some more questions, I guess.
Then I move. Where was I? I was here, I think.
Yes.
Yeah, yeah. So the Aricia Polyommatus group, so also blues, but um, they can be more difficult because the females of Polyommatus, they can also be brown, and that makes it sometimes a bit more complicated. So first of all, the easiest way to identify Polyommatus from Agestis is these two dots. In Agestis, or actually all the Aricias, these two dots are very close to each other. So you get this typical shape wherein Polyommatus, these are rounded, these are not grouped. And once you know this, it makes it very easy to compare Aricia, to distinguish Aricia from Polyommatus. Polyommatus, there are some species which closely look like each other. So I guess this is the most abundant one. You find it almost everywhere, except for Iberia, where it's replaced by Aricia cramera. Um, and I don't even know if you can distinguish them, but at least it helps. Once you cross the border into Iberia, it's normally, when you're south of the Pyrenees, it's cramera. Ataxerxes is this one. So it has much less orange on the upper side than Agestis. It's very dark butterfly. Um, you find it only in northern Britain and Scandinavia. And these two species usually don't occur together. So you either see this one or you see that one. And it's also in Spain, Artaxerxes is replaced by Montensis, which is very much like Artaxerxes, but it's a different species. Here is some maps. So Agestis you find almost everywhere, from Denmark all the way south to the Pyrenees and also in the UK. Cramera is the replacement of Agestis in Iberia. So you find it only south of the Pyrenees. Artaxerxes, northern Britain, and Norway. And Montensis is a more mountain butterfly, like Artaxerxes, by the way. Sorry, I forgot. And you find it in Iberia. Now, the two Polyommatus, Icarus and Thersites, can be challenging. Of course, Icarus always has two basal spots, but not always. So there are also sometimes Icarus without basal spots. Thyzetes never has basal spots, as Amandus never has basal spots. Amandus is really different. It's bigger, and the orange goes not that far. It's only a few orange dots, and it's always near Vitia. So it's almost all my photos of Amandus, I have them sitting on Vitia because that's what the larvae feed on and what they always seem to be neat in the neighborhood. Now, Icarus and Thyzetes can be difficult. In the field, actually, I also put it here. Thyzetes has this very typical different shape of color in the point of the wing. This has to do with the male organs to produce male glands to produce a scent to attract females. Icarus doesn't have that. So Icarus is more or less the same color blue. Thyzetes has this different shape, and I think in this photo you see it a bit better. There are some other characteristics, like the way the black dot and the orange meet up, like you can see here, and there is also this characteristic where in Icarus these three dots are more or less aligned, and in Thyzetes the middle one is a little bit out of the straight line. Now, you will all agree this is really complicated. So these are difficult species. The easiest, and works from a distance, look at the male upper side, if it looks like this or if it looks like one color. That's the easiest one. But it can be a challenge to distinguish them if you have only a photo of the underside. It's doable, but it can be complicated. However, Icarus is everywhere in the Atlantic, but Thyzetes is only in France and in Spain. And Thyzetes is also bound to Onobrichus. There we have the plant again. We have also seen it with Cupido osiris. The species is really always near an Onobrichus plant, which you can see here. The other blues, mainly, well, so this is Icarus, which you find everywhere. You have Belargus, which and Corydon, and they have always these black fringes, black line. In the fringes, you have white and black alternating. And this is, once you know this is really easy. They are also a bit bigger, but it doesn't always have. The females also have this. But for the rest, they are very much like each other, although they have a bit more orange on Belargus. Belargus, well, is almost impossible to catch the real color on a photo because it's in the field, it's very, very, very blue. And this helps to identify the species or distinguish it from Icarus. The undersides are a bit more difficult. The easiest is actually in the females, where the Corydon females are very brown, much browner than the Belargus, which are very nicely arranged, but they still look a lot. So the fringe always helps. It's with black and white alternated. So you find them in most of the Atlantic zone south of Belgium and even in the Belgium. In the Netherlands, these are only migrants. Corydon can be migrating. Large fritillaries, so large orange butterflies. The three main species that you find everywhere in the Atlantic are Adipe, Niobe, and Aglaia. Adipe and Niobe have this line of reddish dots. You find it also here, which distinguish them from Aglaia, which does not have that. Difference between those two species is, first of all, that in Niobe, these veins are black or dark, and in Adipe, they are yellowish or orange, so not so clear. You can also, on the upper side, Adipa has always very clear androconial patches, which in Niobe are much thinner, less developed. So that helps to distinguish those larger fritillaries. I consider the other ones as Paphia as easy ones, so I skip those. The Brenthis species can be difficult, although they are, because they both have this purplish color, because it can also be like this in Ino. The easiest to distinguish the Brenthis species is that Brenthis daphne has two colors in this middle cell, where Ino just has one color, it's yellow. Also, Daphne is a bit bigger. Also on the upper side, it's more differently arranged. In Ino, this is usually almost a black line, and in Daphne, it's droplets of black. These two species don't often occur together. This is typical habitat of Brenthis Ino. It's typically fattened in rough grasslands with lots of filipendula, which is the fruit plant, and it prefers. You very often find it abandoned valleys where they stop mowing, and then there is a period in which there is a lot of filipendula, and then the species can thrive. Daphne lives on brambles, so rubus, and can use much drier habitats as this example. You typically find it in the edge, so where the brambles also grow. So in the edge of the woodland, you can find Brenthis daphne. Boloria species. Boloria selene is a species you can find all over the whole Atlantic. It's the most abundant of the small fritillaries. It has typically a few white spots. That's why, in comparison to Euphrosinae, which only has one white spot in the middle of the underside. Still, Selene usually has more than one generation, so you can also find it in summer, where Euphrosyne usually just has one generation in spring. So in spring you can find both species, but not so often mixed up. Euphrosyne is more a species of woodland edges, where Selene is more a species, well, it's in a lot of habitats, but very often it likes, for example, humid grasslands, but also in humid forest edges you can find it. Boloria is especially a species of dry habitats. It has very dark and big black dots on the upper side, and the underside is very purple. It's typically a species on very dry areas, but also can come onto agricultural fields, which are left alone for 1 or 2 years. Then Boloria can colonize those. The last one, Boloria aquilinaris, I added it because it's an Atlantic species, but you only find it on peat bogs. And it's one of the few species which you can actually see on peat bogs. So it's very different from the others. So this is typically Boloria aquilinaris habitat, in this case in the Netherlands, where you can see that these are areas that are not just difficult to go onto, but it's also quite dangerous. You might disappear and never be found again. Then we have the other small fritillaries. We have a few. Athalia is really the widespread and most abundant Melitaea that you can find. It's in almost a whole of the Atlantic zone. It has this typical white form of these marginal areas, and there is this stripe. Diamina is another species, but it always has these black dots in this orange band, and a much clearer orange in the edge. The top is also different. I think I forgot to include a photo of the top. Yeah, because the top of the upper side of Diamina is very dark. It's much darker than the other Melitaea species. In France and Iberia, you find Partenoides, which has a the upper side is a bit differently arranged. It always seems to miss this black line, which is present in the other species. Dejone, well, it's on the edge. You will find it maybe in parts of Spain. It has always a bit more colors in the upper side. It's never uniform orange, which others can be. And also the underside is very neatly arranged. What also helps, this species has two generations. This has two generations, so it flies in May and then back in August. Athalia and Diamina have just one generation. Usually in the end of June, beginning of July is typically the moment you can find these two butterflies. That also helps to identify them. So Athalia is widespread. Seladusa replaces Athalia, by the way, in southern France and Iberia. Partenoides, widespread in France and Iberia. Diamina is actually not very common and widespread. It always is in habitats with Valeriana, and Dejone is in southern France and Iberia. Oh yeah, here I have some maps in which you can see. For example, Athalia is really rare in UK and the Netherlands. It's just at a few populations. It's also a bit on Denmark and Norway, and then in France. Seladusa is only in the very south, replaces the other species, replaces Athalia. Partenoides can be very widespread in parts of France and northern Spain. And Dejone is especially also in the same area, but it's rarer than the other one. This is what I call the Melitaeas didima. That's this very clear orange band, and here also a very clear orange band, which is very typical for this species. The males are very, very orange, and that also helps to identify them. Xynxia is the only species with always black dots in this band, which you find here. You see it on the upper side and on the underside. Phoebe can also have some dots, but never as clear and never complete set of dots as Xynxia has. What's typical for Phoebe is this large, I think it's cell number 5, which is clearly larger than the rest. It stands out. Here it's also a bit bigger, but never as big as in Phoebe. On the underside, this band has always orange dots in them, which helps to identify this species in the field. Phoebe is also a bit bigger than the other two, but as, again, size only helps if you see them next to each other. We're running a little bit late, but think I'll just need 5 more minutes. The Lasiommata species, Lasiommata megara, is the most widespread species in the southern and middle of the North Atlantic. It always has this orange band here, because that is missing in Meira. These are two very black specimens, but they can also be much more orange. But they never have this orange band on the hind wing, which you find in Megara. Petropolitana is the last species. It's smaller and you only find it in the Atlantic in Norway. So this species is not in the rest of the Atlantic. It's a bit smaller and very grayish, always dark. Here's some maps. So Megara more or less stops in Denmark, south of Denmark. Megara, I've always wondered. I've never paid attention when I was in Paris, but it seems to be quite abundant in Paris because I think this is Paris. Anyway, and then the rest of France, Petropolitana in the Atlantic zone, only in Norway. The Coenonympha species almost there.
Chris, before you go to Coenonympha, there is a question of Melitaeas. Sorry.
Yeah.
How could be distinguished among Parthenoides and Natalia?
There are a few things that help. First of all, Athalia flies in June, July. Parthenoides flies in 2 generations. So typically in May and then again in August. But yeah, I should have included a photograph of the upper side of Athalia because then we could compare. But in Athalia, usually this line, there is a line here, which is now missing. Here it looks like these cells are a bit larger than the other ones. And that's something which, in my experience, helps a lot to distinguish Parthenoides from Athalia or Celadusa for that part, because I think Athalia and Celadusa cannot be separated only on the place where you are. But so this is, if you go in June, July, you always find Athalia and just some old specimens of Parthenoides. That also helps. So, because sometimes they can be really difficult. I mean, this is a beautiful specimen, but sometimes, of course, they are a bit worn, and then it is difficult to. I find it difficult to distinguish them. So.
Okay, thank you. No more questions.
Okay. Yeah, the Coenonympha, whatever. We have a few species which are interesting. So Glycerion, or actually in Iberia, it's Ephiurides. It has this orange band at the end here. You can see that. This is the Iberian form. This is the continental form, but there is also this orange band along the and of course, it has some of the dots. In Iberia, they are really big, but in continental form, they are smaller and have this white patch, which you find here and which you find here. Of course, the white patch is also in some other species like Thulia. I come back to Thulia because Thulia is can also be identified on the habitat. This is always on peat bogs or very wet grasslands. Pamphilus is everywhere, has no real markings. It's more brownish, although it can have some dots at the end. But Argynnis has clear, large dots and one which is on the other side of the white band. It has a very clear white band, big and pronounced. And one, this is always on the other side. Oedipus is very brown, completely brown, actually often looking a bit more like Aphantopas hipporanthus, and has these row of black dots, and one is displaced, and the females have on the underside of the forewing a whole row, and which is in the males is almost missing, much smaller. But what also helps is the habitat. I told you, for Thulia, you are always in a situation as this, wet grasslands or wet heathlands or peat bogs, where you can find this species. You're always in a habitat which looks like this and is very wet. Where Argynnis you can find actually in this typical landscape. So rolling hills with some bushes and some small forests and lonely trees. Typically on places like this is where you would search for Argynnis. They always are near to trees or bushes. Oedipus is also a wet species. It usually is flying on this type of wet habitats, which are also not as wet as Thulia, and Thulia is mostly in the north, where Oedipus is in the south of the Atlantic. Usually, if you are here, you meet a lot of mosquito female friends which love your visit. And yeah, it's always a pleasure to be there, but you're also happy to leave. The large brown Satyrids, so these are really big butterflies which fly typically in the height of summer. Brintesia circe has this small extra white patch which is missing in the Hipparchia species. This is the most abundant one, Brintesia circe. The other two, yeah, in France you can have both of them, and they're really hard to identify, I think. Officially, you have to check for to squeeze and look at their genitalia or some structures around there. All the other ways to look at DNA. But in Spain, it's fairly simple because in south of the Pyrenees, it's only Alcyona or Hermiona or whatever name. It has that problem. It has several, it's not always clear. Here it's called Alcyona, but it's also often called Hermiona. Fagi is the more common species in France. But it's actually also interesting that so here you see that Circe is very abundant in the southern half of the Atlantic. Fagi also. Hermione is in the Iberia, plus local in northern Germany, and it's just not on the map, but also in Denmark. So it has some very remote other populations. Hipparchia's Semele is another species. You find it in the Atlantic, especially in these heathlands or dunes with a lot of sand. And Arethusa is mainly found in France and Iberia. This is more a grassland species and Hipparchia, but in the south, Hipparchia also can be found in other places. Look at the shape of the white here. In Hipparchia semele, this is always like this, very clear usually. In Arethusa, it's much more like a straight or rounded white lighter band. And the two most abundant species we have, together with Melanacher galatea, are Tithonus and Manioleurtina. I do them because sometimes people find them difficult, but actually the best is to look at these white dots. In Tithonus, you find small white dots. In Urtina, never. They are, if you find dots, they are black. Also, this is often pointed out, but we know this is not always correct. So Tithonus usually has two white dots and Urtina just one, but there are also Urtinas with two dots. So it's not an absolute, but 95% of the times it works. And this is Tithonus with two and Urtina with one white dot. So I'm more or less managed. So this is again the guide, which I can only advise because many of the photos you have seen come from this guide, and you can Google it and find it, or send me an email. I can give you a link. So it's a free download, and it's really, I think, a very helpful way to use a helpful book to use. And I'm very thankful for Peter Gargeli to allow me to use the photos also in this presentation. I think that's it. 10 minutes over time, but still, we manage. More or less.
Great. Yeah, thank you so much, Chris. I think it was really great. I put already the book link on the chat,
Okay.
so everyone could have it.